To better understand this subject and to help us appreciate the importance of Dr. Strecker's work, I'm going to briefly clarify and emphasize some of the information in this presentation. Virology is the study of viruses. It deals with tiny living organisms visible only with the use of the most powerful electron microscopes. In fact, hundreds of thousands of AIDS viruses can easily fit on the head of a pin. What makes the AIDS virus particularly deadly is its ability to not only invade and neutralize human cells, but the virus's ability to put its own genetic material inside the human cell's genetic structure, thereby allowing the virus to use the human cell as a kind of virus factory, reproducing more viruses from the human cell's raw materials. Unlike larger organisms like bacteria, viruses do not respond to conventional medical treatment, much like the common cold virus cannot be treated effectively by drugs.
Now, how does this AIDS virus, which is a human retrovirus of the D type, how does this virus affect humans? Basically, the immune system of humans is broken down into two parts. It's very simple. One is called B cells and one is called T cells. B cells are derived from the bursa fabricius, but the easy way to remember B is that they control basically production of antibodies and control bacterial infections, therefore B rather than bursa. You can think of it as bacteria. T cell systems control opportunistic infections such as pneumocystis carinii pneumonia, the production of cancers like Kaposi sarcoma, and other microorganisms such as tuberculosis. So that if you wiped out the T cell system, you could see the arisal of opportunistic infections such as pneumocystic carinii pneumonia, a hallmark of AIDS, Kaposi sarcoma, an alleged sarcoma, and another hallmark of AIDS, or other diseases. If you wiped out the B system, you would have trouble protecting yourself against bacteria and perhaps in developing antibodies. Now, in the case of the AIDS virus, once it infects the human body, selectively destroys the T4 cells of the human body. The T4 cells are a division of the T lymphocytic system, the one that helps us control cancers, fungi, pneumocystis carinii pneumonia, and other organisms. The AIDS virus selectively leads to the destruction of those cells. If you look at the overall incidence of these new so-called human retroviruses, you'll discover the following. First, there is HTLV1, a new human retrovirus which is responsible for T-cell leukemia. There is HTLV2, which has this appearance, which is responsible for the development of hairy cell leukemia. And we have HTLV3, which looks like this. These are the best known so far, which is the AIDS virus, responsible for AIDS. Now, interestingly enough, when you put these viruses into tissue culture, what happens? This virus is proliferative in tissue culture. It makes things grow. It's not surprising, therefore, that you might see the arisal of a T-cell leukemia because the virus's very nature is to make the cells proliferate. This virus is proliferative in tissue culture, results in humans in the development of hairy cell leukemia. And again, by its basic proliferative nature in tissue culture, it's not surprising that you can see arising hairy cell leukemia. This virus in tissue culture is destructive. It was one of the reasons that they had trouble getting enough of the virus because suppose this was a tissue culture, every time they put in the AIDS virus, what would happen was they'd come back in a few days and the tissue culture would be dead. Basically, there would be left just debris with very little virus, no living cells. That is basically what happens in human beings because in humans, we are nothing more than walking tissue cultures. So, suppose we have a human being here. He gets infected with the AIDS virus, and what happens? Eventually, the AIDS virus wipes out his T4 lymphocytes, destroys his thymus most probably, and as a result, relieves him immune suppressed, immune compromised, and susceptible to the development of infections such as pneumocystis carinii pneumonia or the arisal of cancer such as Kaposi sarcoma. These have been the basic hallmarks of the AIDS virus. But again, let's look at the overall picture. And the overall picture is that suddenly in humans we have an explosion of disease, not just AIDS, but other retroviruses, HTLV1, a T-cell leukemia virus, HTLV2, HTLV3, HTLV4, which is a new, recently recognized AIDS virus, also known as HIV2, HTLV5, which is the cause of mycosis fungoides, and HTLV1 lookalike, which looks like this. I call it 1 lookalike. Now, even looking at, say, let's throw AIDS out for a minute, you have to ask yourself, where did all these other viruses come from? And my explanation as to where these other things have come from, along with the AIDS virus, is the following. If you look at animals, particularly cattle and sheep, you'll discover an interesting phenomenon. In cattle, there's a virus known as bovine leukemia virus, which has the exact same morphology, which means shape, the exact same relative molecular weight, the same magnesium dependency, and the ability to produce B and T cell leukemia of cattle, and is proliferative in tissue culture. If you look at bovine syncytial virus, you'll find another virus of cattle, which has the same shape, the same magnesium dependency, the same basic appearance, and produces hairy cell leukemia in cattle. If you look back, you can discover a virus known as bovine Visna virus, which has the same appearance as AIDS, the same molecular weight, the same magnesium dependency, and in 1974, either 1 or 3 here, bovine leukemia or bovine Visna virus, was producing pneumocystis carinii pneumonia in chimpanzees. And if that isn't AIDS, I don't know what it is. Now we have HTLV4, which may represent a recombination between Visna and HTLV2, or bovine syncytial virus here, which is a new AIDS virus, growing, just identified, which has this appearance. We have HTLV5, which I'm not sure in animals what it is, and we have HTLV1 lookalike.
You'll recall that Dr. Strecker has shown us that the AIDS virus selects and destroys T4 cells. These are the cells in our bodies that protect us against the development of cancers. Now, persons infected with the AIDS virus have T4 cell destruction and subsequent development of specific types of cancer. These include Kaposi sarcoma and pneumocystic carinii pneumonia, which are fatal. Looking at the overall virus picture in a general way, Dr. Strecker has pointed out that there are several other deadly retroviruses, besides the actual AIDS virus, which are infecting humans and causing cancers, including cancer of the blood, leukemia. Dr. Strecker makes an interesting and startling correlation. These human cancer-causing retroviruses, including AIDS, all have striking similarities to animal viruses, but not from the green monkey, from cattle and sheep, known as bovine and Visna viruses. Now, the implications of these similarities between human and animal viruses is disturbing. How did animal viruses get into humans? As we shall see, this subject begins to reveal the true origins of the AIDS virus.